Thank you very much, Glenn. It's a pleasure to be here, and thank you for being here. It's 15 years now since human embryo stem cells were first isolated. Some 35 years since a comparable achievement in mouse that led us to where we are today. And rightly so, and as you've heard from Andreas's talk earlier, over the last half a dozen or more years, there's been a lot of progress in terms of creating the structures that are needed to transform the science into an industry that is ethical and that is controlled and that is capable of giving tangible rewards. I'm going to speak to you today about the European Bank for Induced Stem Cells, which constitutes another of those efforts to try to create a standardized banking structure in relation to induced pluripotent stem cells. So harking back to our school days, today's lesson is about an introduction to Ebis and what it's done so far. And specifically, I will be talking to you about an effort that I've had a hand in leading relating to the establishment of a hot start process to put the wheels on the bank. Then I'm going to take a sidestep and give you or speak briefly about a perspective on what all is involved in the industrialization of this resource and remind us of some of the assumptions that we carry and some of the things that we need to do going forward. And I will end with an example of a project that I have an association with. So, the Ebis of the mission, the mission of Ebis, not the Ebis of mission, is to create a standardized human-induced pluripotent stem cell bank that has made an effort to standardize the key essentials of procurement of tissue from donors, its banking, associated quality control, and then the distribution of this resource. At a glance, this is a consortium, I knew the number at one point, but approaching 30 members, over half a dozen pharmaceutical partners listed up there in purple, led by Pfizer, Nucensis, and specifically Dr. Tim Alsop is the coordinator. The managing entity of the consortium of the non-FPA partners, FPA stands for European Federation of Industrial Partners, is the University of Edinburgh and Roslin Cells, a company of which the university is a stakeholder. And it has been created and strengthened by association of academic centers that have been engaged and leading at the forefront of induced pluripotent stem cell biology and usage in Europe, and other centers such as the Wellcome Trust Sanger Center that has its own efforts to create a large bank of induced pluripotent stem cells, and it's associated with other efforts to use this resource, as Jim will have spoken to you about, in the form of Stem Bank. In the first instance, we started this in January 2014, and the goal was to become as proficient and as operational as quickly as possible. And so we wanted to both learn through experience of doing and establish a foundational collection and catalog. The first element of that catalog was what we refer to as the hot start process, where the consortium agreed, first identified amongst the centers that had been actively engaged, academic centers actively engaged in human-induced pluripotent stem cell derivation. We identified lines that they had established and characterized that would be of interest to the European community. We agreed as a consortium standardized protocols for the expansion of these cells under feeder-free conditions, their cryopreservation, their distribution. We agreed the principles of consent that we would seek to confirm that these lines were collected or procured from donors for. And with this, then, we decided that we would create this hot start collection of lines, and the process of expanding these and then putting them through a quality control and through to distribution would give us the beginnings of an operation that could subsequently be matured. Key also in this effort was aligning with other established efforts to establish large collections of induced pluripotent stem cells, most notably that going on as part of the EBiSC program funded by the Wellcome Trust to create induced pluripotent stem cell lines representative of the general population. And our ambition then to benefit that program by helping with the distribution of these lines, banking and distribution of these lines outward. As well, there was a pot of money made available within the program to commission the production of new induced pluripotent stem cell lines once we could identify where the gaps were. And this also included the using of this fund to create genetically modified lines. As isogenic controls where specific gene polymorphisms might be altered, associated with disease might be altered. And then ultimately other sources, such as those already funded by other running in European Union-funded programs. So these collectively constitute the foundational collection and the broader catalog. And this is another way of depicting what I've just communicated here in terms of identifying the representation of induced pluripotent stem cells in the catalog from the founding academic centers, as well as other programs that, pardon me, are already existing or either within Europe or outside of Europe, in this case here, the United States, and with Ebis positioning itself in the center to facilitate the subsequent distribution. So, the hot start. As I said, this was about getting the wheels on, establishing best practice through experience. I've already alluded to how we defined the principles of procurement that we wanted to see, the terms of consent. One of the key terms of the consent, but not the only, I assure you, term was the fact that there would be no commercial restrictions on this program. I forgot to mention a very important point, that this program is funded and supported by matching contributions from the European community in terms of cash and the FP partners in terms of cash and contributions in kind. And clearly, although the resource is to benefit Europe and the world in the first instance, the FP partners were very concerned, and understandably, for the assurance that the lines that would be deposited and made available through this resource would be something that they could access. And build and incorporate into their drug development pipeline, which of course is a commercial activity. Building on strengths, the HESREG or the HPSC reg that Andreas spoke about, central to the initiative in terms of being the starting point of the information management system to collect the data associated with these lines. And as I indicated also, the aspiration to standardize lines that may have been, the production of lines that may have been derived under and expanded under different culture conditions in the first instance. Our preference would be to have a standardized approach and not need to try to accommodate all of the variations on how these cells can be cultured that exist in the field. So the hot start process then, again, pictured a different way. Procurement, In the first instance, for the hot start, we were using lines that had already been derived, for which then we defined what are the terms of the consenting process that are agreed to be an important standard, and checking that the lines that were procured as part of the hot start collection were obtained in that manner. Again, means to assure the capture of donor data, anonymized donor data that would then be linked to these lines, because the lines in themselves, quite frankly, are only the small part of the picture. It's the data-rich clinical, the opportunity to access and have ability to connect patient history information, clinical information with the lines is the key to the resource. The lines then were to be expanded by standard protocols, which were reinforced through training at the EBiSC, again, taking advantage of another established resource. And the National Institute for Biological Standards Control, of course, being the, if you will, the controller and the developer and the controller of the UK Stem Cell Bank, with a long-standing experience in both banking and training. And our feeder-free protocols then were defined following a survey of what was being used by the partnership, the consortium, and others. And we essentially identified two options of feeder-free culture based on either M-teaser or a minimal essential 8 media, and two options for substrates that could be used, and an agreed passaging regime involving use of calcium or divalent cation chelator. We accompanied all of this with guidance documents as to how the cells should be harvested, after harvesting and for cryopreservation, pooling of the populations to be aliquoted, and keeping track of which the order of the filling of the vials, so that subsequently one could check the resource by evaluating vials at the beginning, at the end of pooling for cryopreservation or aliquoting, and if one identified difficulties with a vial, being able to work inward to establish at what point cells may have become compromised. A lot of effort went into establishing a label that was both human readable and machine readable, to then be able to be implemented in an automated cryopreservation system established at the Fraunhofer IBMT. And again, data being harvested, collected, deposited with HESREG if it was not already. So that was all going on then at the derivation centers that created the lines, with central support in terms of guidance documents agreed by the consortium to be implemented. And then at the central facility at Roslin Cells, we would engage in a systematic, documented registry of the progression of that resource, with the batch records and standard protocols. We would then engage in a very finite and pragmatic quality control that was about very much of a banking mindset, not an information-rich research effort. And so our banking was about freeze-thawing, confirming viability, doing genetic PCR-based, microsatellite PCR-based identification of microsatellite markers to track identity, G-banded chromosome confirmation of genetic integrity, looking at morphology, culturing under antibiotic-free conditions, which we insisted upon that the derivation centers did, so that if there were any microbiologicals, they would become manifest in our hands as opposed to the hands of the end user once they were distributed. Also PCR testing for mycoplasma virology, etc. And as regards assessment of pluripotency, In the first instance, we have relied on the data that has come from the derivation centers, but any new lines being commissioned would undergo a very minimalistic assessment of pluripotency markers by qPCR and flow cytometry as quantitative approximative is that a word? Indices of pluripotency. Meanwhile, at EBiSC, there is a development of a embryoid body differentiation-based assay in order to engage in some type of quantitative and predictive measure of lineage potency without actually putting cells through all manner of differentiation protocols. All of this data is collected in certificates of assessment of assurance, I should say, and then this resource is shipped to our mirror bank and distribution centers with a minimalistic contingent of a stocked line being held at Roslin cells. So that's what it looks like. This is to reiterate the significant effort that my colleagues, not me, put into establishing a label that was both informative to an individual picking up a vial, but then able to be traced to other reference tracking identities that had been associated with the line and machine readable, so it could be implemented in Fraunhofer's automated cryo-storage, controlled cryo-storage system. So I started by saying the aspiration was to create a resource that would underpin an industry. And I think that I should have persuaded you by now that indeed this is what has taken place. And so why would you come to Ebis as a source of stem cell lines as opposed to your friendly neighborhood neighbor just 2 doors down who's just willing to give you a plate of cells and run away and do your experiment? Because of the heavy documentation that actually says and warrants that what you have is what it says it is on the label, and backed up by the prospective access to training protocols to be able to work with that resource so that when you thaw it, it will survive and you will get meaningful usage out of it. A very fair question is to ask, well, how did it go when you were trying to get these centers to all work to the same, sing to the same hymn sheet? Indeed, as we all know, organizing scientists is a lot like herding cats. And so it's a fair question to ask, to which I give you some metadata now of that progress. So we had 7 centers. And the table here coded in green for job done, yellow for, well, we almost got there. Red would have been dangerous. Across these different quadrants. So all of the centers pretty much delivered on their specified contribution, identified a contribution, sent lines to the central bank, all or most of them. As we start moving down, we start slipping notches. Completion at this point in time now, we are a year and a half into the process. We still have a few outstanding depositors forms to collect and testing forms, and nailing down that patient information and consent, which we were assured was, as we said, was aligned with our terms. Of course, this is why we did all of this, so we could learn what to do, what works, what doesn't work. We still have a few outstanding material deposition agreements to be signed. And going forward, certainly before we accept lines into the bank and a lot of people start putting their shoulders to the wheel, we actually get the agreement sorted out first and then we work up from there. but still, I think a positive experience. And how about working with cell lines that had been derived under different conditions and expanded under different conditions and of different cell sources? How were these feeder-free culture conditions protocols? How effective were they? Well, pretty darn good, actually. And again, another reason why you would want to be able to access our lines and the protocols if you are entering into this field. Because of the 47 lines that we've had so far, good majority of them recovered well on thawing. Morphology, about half of them had the consistent morphology that you would want. Most of them had little level of differentiation. Sterility issues, the majority were sterile. At least there were no microbiologicals growing after 3 passages in antibiotic-free media. And we did encounter a cell identity issue where lines were actually on doing microsatellite PCR, not the lines that had been supposedly sent to us. And again, this reinforces why the measure of looking and tracing identity is important. What have we learned? We learned along the way that despite the best will in the world, depositors will have concerns about how rapidly this resource gets into the hands of their competitors. We have learned that our feeder-free conditions, not ours, those that are being used by the field, actually are pretty darn robust. That's good news for the commercial distributors thereof. Training is absolutely essential, and the centers that did the best are the ones that turned up at the training sessions and participated fully with the process. Biosample ID label was, I think, a tour de force. I'm sorry, I keep playing that up, but it was a lot of work and it was well done. We've established an excellent interface with the European Bioinformatics Institute and HESREG, and which is growing. And we've now put in place a laboratory and management system. So from the ground up, there's an electronic registry and control of what's being done, monitoring of what's being done. And we've also come across member-specific restrictions in the access to IPS cell lines created in their country. So in Spain, it turns out that there are nationally legislated requirements that preclude the ability of anyone other than a Spanish institute distributing directly to an end user an IPS cell source. We also have some standard IPS cell lines originating from Cologne and Bonn that could one day perhaps become the new Wisconsin Y-cell H9 and H1s that we hope would be taken up by the field. Roslin Cells has also created a line as well that is available through the bank. I've given you information. Now I'm going to change gears on you and ask us all to take a step back and look at some of the presumptions that we're working with when we aspire to use this wonderful resource. We are presuming cell and tissue functional maturity after we've differentiated these cells in a dish. We are presuming physiological and disease relevance of a phenotype that we might have, which negates, of course, the fact that disease is a mix of nature versus nurture. The environment has an effect on how a disease manifests clearly. How do you reproduce that in cells in a dish? We should ask ourselves, what is the practical value of what we're doing? Not just, we can do it, so let's do it. What do we see, can we convincingly convince ourselves, not just argue, convince ourselves is the value of the effort? And there's lots of places to spend our money in this world. There's a whole third world that is dying of diseases that, quite frankly, we take for granted. So can we do a cost-benefit analysis that this is where we should be sinking our money? These are some quick answers to the presumptions that we have, which we're all aware of, but are worth voicing. We know that these differentiated cell types that are being created actually have more of an embryonic or fetal resemblance than they do a adult resemblance in phenotype. We know that there are factor codependence issues associated with disease manifestation, how the environment interacts, aging, etc. When it comes to value, we need to be clear on whether we're trying to define a prospective, retrospective, or corrective value. And this applies the last to the idea of cells for therapy as well, which Ebis is not concerned with. It's concerned for cells for discovery. And what is the efficacy and affordability of current procedures? Well, they're pretty darn expensive. This is, if you compare what we spend in the life sciences to other industries, sometimes the numbers can be eye-watering. Here's another question. When we say that we're going to represent diseases in a dish and person-specific representation, disease representation, we should be asking ourselves, which of us is representative enough? And at what point in our life is someone representative enough? Does it really matter when we collect these cells? I love this mural, by the way. Those of you who've heard me speak will be familiar with this slide. It hangs in the south terminal of Gatwick, and of course it's a lovely picture of the Queen, a mural made of Polaroids of British citizens, which speaks to the point that the Queen is representative of Britain, but nobody for a minute would suggest that simply studying her, iPS cell lines from her will indeed represent the UK population. So I'm being a bit facetious here, but I make my point. I think I've made my point. So if one individual or two or three, or heck, why not just say half a dozen, which is at the present time, getting published in Nature and Science requires that you represent a phenotype in a dish from numerous, donor-derived iPS cell lines. So, but is that enough? Well, strictly speaking, no, it's not. And so what do we need to do? We need to drive the cost out of this process. And if you look at any other field, whether it's DNA sequencing or the cost of making clinical-grade cell lines, price comes down. I have the T-shirt myself of making clinical-grade iPS cell lines. The first lines cost well over £500,000. Why? Well, basically, you take the size of the grant I got to do the work and the number of lines I created, and that's how you end up how much it costs to do the first lines. Of course, today, if you go to the Ebis, it will probably cost you about £1,000 to access a heavily quality-assured iPS cell lines. It'll take more to make it. And really where we need to be is the £1,000 iPS cell line, donor-specific, so that you might be able to plug into its usage as a clinical diagnostic to help people decide, what is the best treatment option for them. Now, it's a short talk today, so I'm not going to dwell in, what you might do for that. You'll have to invite me back another day for that. There's also another consideration, and that is if you can make iPS cell lines for everybody, well, then how the hell are you going to work with all these iPS cell lines? And that requires an industrialized outlook to screening, which, of course, the pharmaceutical industry is quite adept at, and there are robots that move things around. But we do need to build those further to the point that technology is not just in the hands of the pharmaceutical companies. And by way of example, and I am certainly biased in my presentation, but what the hey, there are projects like the one that I have the good fortune of being associated with, and a sister company to Roslin Cells, Roslin Cell Lab, led by Professor Heiko Zimmermann at the Fraunhofer IBMT, funded by the European Union, known as DropTech. And DropTech is about creating an automated human-induced pluripotent stem cell variation of the only, to my knowledge, embryo stem cell independently validated embryo stem cell-based toxicity assay, that being the MEST test, which was developed quite a number of years ago, actually now, I dare say probably a decade ago. And if you're interested in its formalization as an accessible protocol, I refer you there to Nature Protocols. So what does that assay look like? Well, originally, by hand, it involved a hanging drop, a stage of supporting embryoid body differentiation. These drops of media would be put on the underside of a lid of a dish, flipped over, and then after a period of culture, these would be allowed to adhere and spontaneously differentiate into masses that would contain cardiomyocyte-like cells that would then beat and pulse. So the exposure to prospective toxicants would occur at the level of undifferentiated cells, and then the cells would be put through this differentiation protocol. And prospectively, the drop-tech automated hanging drop approach then would automate this step of creating drops containing embryoid bodies that would perhaps, if there was a need for a adherent substrate to get terminal maturation, one could achieve by adding in a substrate in the form of beads that the cells could adhere to. And then afterwards, you would either he steps back in time, either see beading in the drop itself, or you would have attachment to a substrate and beading. This is the kind of robotic equipment that actually, in the grand scale of things, is not as expensive as other pieces of kit. In the tens of thousands, nano drop plotting robot, and we essentially have a feeding dish and a dispensing dish. This dish is where the hanging drops would be made. This is a zoom in of the underside of this plate where you see the drops hanging. And here we have, this is provided kindly by Ina Meiser at Fraunhofer IBMT. Adding in the alginate beads then allows the cells to attach to these beads. And then, if I am fortunate, and I may not because, there we are, there we go. If this is kindly provided to me by Dr. Rini Kutsuraki, a former PhD student of mine, now working for Roslin Cell Lab. You can then, this is human-induced pluripotent stem cell-derived iPS outgrowing, attaching to a substrate or to the plate and doing the lovely beading thing that makes for such great home movies. And then lastly, in the way of videos, again, I'm entertaining here, so bear with me. I hope you aren't being entertained. In terms of actually dispensing the drops, we then have a TCAN robot. Now, this is an expensive piece of kit that would, you see here in action, essentially picking up fluid and then creating your drops into which, with the different pipetter, the embryoid bodies or cells that would then form an embryoid body would go on to coalesce. Very good. Right. So what did that nice man, Paul de Sousa, tell me today? I've told you about Ebis, which has been a fantastic effort that I'm very pleased to be associated with. That's all about trying to standardize the procurement, the expansion, the banking of induced pluripotent stem cells to support an industry as well as the global research community. There are iPS cell lines now available, and I refer you to the website to procure them. And I've also, by way of an example, given you some nice images and pictures to think about as an exemplar of what needs to happen going forward to be able to use the iPS cell lines that are being created. I'd like to thank a large number of people that I essentially sum up in two words, DropTech and the Ebis Consortium. A picture speaks 1,000 words, and here is the Ebis Consortium saying hello and thank you to you all. Thank you.